A very good morning to all of you. So last time we studied about the plasma membrane, cytoplasm and nucleus, which are three components of cell present in all the living organisms, or which is present in the cells of all the living organisms. Today we will study about cell wall. Now you can see the image also. This is the image of the plant cells. What is cell wall? Cell wall, it is present outside the cell membrane, and it is found only in the plant cell or in cells of certain bacteria. It is rigid. It is rigid means it is tough. So what is the advantage? That it provides strength to the plant, and it also protects the plants from outside harsh environmental conditions. And this cell wall is made up of cellulose. What is cellulose? Any one of you can answer? Any one of you who knows the answer can answer by using microphone. Okay, cellulose is actually a complex carbohydrate. Now students, if this cell, plant cell, if you keep it in a concentrated sugar solution is what? It is hypertonic solution, where the sugar is more and water is less. So if you keep a plant cell in such type of solutions, what will happen? Now the cytoplasm, outside is cell wall, then is cell membrane, and then is cytoplasm. And in the center of the cytoplasm is present nucleus. And all the organelles, the cellular structures, they lie in the cytoplasm. So when we keep a plant cell in a hypertonic solution, you know that it shrinks. But actually what happens? Now hypertonic solution is what? Where sugar is more, water is less, as compared to cell. So in such a case, the cell loses water. The water from the cell comes out in the surroundings. Okay, this is osmosis, because it is a movement of water from the cell to the surrounding through plasma membrane. So in this case, children, now what happens? The cytoplasm, actually it is fixed to the, or it is fixed to the this thing, your cell wall, or it presses against the cell wall. Now what happens? The cell wall, the cytoplasm, it starts detaching from the cell wall. It starts shrinking, and the cell also shrinks. You have seen in resins also. Now if this plasmolyzed cell, if you keep in a hypertonic solution is a dilute solution, or a solution where sugar is less or water is more. It is not sugar, it can be salt also. So that is a hypotonic solution. If you keep this plasmolyzed cell in a hypotonic solution, hypotonic or dilute solution, what will happen? Again, the water from the surroundings or from the solution will move inside the cell, or you can say osmosis will take place. There will be a movement of water through plasma membrane, and again the cytoplasm will press against the cell wall and cell will swell. This is called deplasmolysis. Okay, now activity 5.6 that you can read, it is how you will prepare a temporary mount of a leaf and see the view it under the microscope. So you just read it, afterwards we will discuss. Question number 1, you can see that is a homework. Now children, there is an image of an animal cell. Okay, image 2. Now what are cell organelles? I have told you in the beginning only that the cell organelles, the structures present in the cell are the cell organelles. I told you that in the cell also certain structures are present which is smaller in size than the cell, and those are called as cell organelles. What are they called? Cell organelles. Generally, most of the structures are membrane bound, means they are surrounded by a membrane. Only in case of prokaryotes, the structures or the cell organelles are not surrounded by a membrane. Okay, children, while explaining you, if you have any doubt, you can ask. You can use your microphone to clear your doubts. So, now first, now why the cell organelles are membrane bound? Just to keep its contents separate. Each cell organelle or structure has some function, and it has certain things inside it which are called as the contents of the cell. So these cell organelles are membrane bound to keep themselves separate from other organelles. Okay, or to keep themselves from other organelles, or external environment. External environment is that only, that to keep itself separate from outside environment, okay, or other organelles. Now these organelles, they perform very much complicated chemical reactions to support cell. Cell supports a cell. It is the living entity, because why we are living, it is present in the living thing. It is the basic living, a fundamental thing, living thing which is present. Now what these organelles are surrounded by the membrane because they perform very complicated chemical reactions, and it is necessary to remain separate from other organelles. Except in prokaryotes. In prokaryotes, children, what happens, the organelles are not surrounded by membranes, but in eukaryotes, the organelles are surrounded by membranes. So this is, now next time I will not say certain structures are present and all. No. I will say cell organelles. So they are the structures which are present in the cell. Now we will study some of the cell organelles in detail. First is endoplasmic reticulum. Endoplasmic reticulum is also written as capital ER. You can see it in image 2, that is of animal cell, and also in image 3. In image 3, you will see the detailed structure of endoplasmic reticulum, and here you will see in the animal cell. It is also present in the plant cell. Now, what are these endoplasmic reticulum? Endoplasmic reticulum is nothing but they are the tubes which are membrane bound. Membrane bound tubes means they are having membrane is surrounding them. All the organelles I told you are membrane bound structures, so they are membrane bound tubes. Okay, and it sometimes appear in the form of long tubules, sometimes in the form of round bags. Okay, it is long tubes. It is similar to cell membrane. Okay, long tubes which are bounded by membrane, which is you can see in the image 2. Okay, now there are actually, children, 2 types of endoplasmic reticulum. How many types? 2 types of endoplasmic reticulum. One is rough endoplasmic reticulum, which is also written as capital RER, and another one is smooth endoplasmic reticulum, which is written as SER. We will study first rough endoplasmic reticulum. Children, rough endoplasmic reticulum, rough. Okay, when you will see it under the microscope, you cannot see it under compound microscope. You need a highly powerful microscope to view it. Okay, so rough endoplasmic reticulum, when you view it under a powerful microscope, it will not look smooth. You know the meaning of smooth? So it will look rough. Okay. Why it looks rough? Because in rough endoplasmic reticulum, there are certain very, very small structures called ribosomes. These ribosomes are attached. What are attached? Ribosomes. So that is why it gives rough appearance. You can see it in image 2. And image 3 also. Okay, image 2, it is in image 2 only the endoplasmic reticulum is shown, and but the detailed image of endoplasmic reticulum you can see in image 3. Okay, in image 3 you can see rough endoplasmic reticulum also, and you can see smooth endoplasmic reticulum also. Now, rough endoplasmic reticulum is what? The endoplasmic reticulum where the ribosomes are attached. Now, ribosomes helps in the synthesis of proteins. Okay, so ribosomes are, what is the main function of the ribosomes? Which are helps in the synthesis of proteins, these ribosomes. And now what happens? These ribosomes, they are attached on the endoplasmic reticulum. Now what is these ribosomes? They synthesize proteins, but these proteins are sent to the different places of cell. Have you understood? Now smooth endoplasmic reticulum. What is smooth endoplasmic reticulum? Smooth endoplasmic reticulum is that where the ribosomes are not attached. Okay, now what is the main function of smooth endoplasmic reticulum? It helps in the manufacture of fat molecules, helps in the manufacture of some type of lipids. Proteins are also manufactured by smooth endoplasmic reticulum, and it helps in the building up of cell membrane. What helps in the building up of cell membrane? Lipids and proteins, because cell membrane is made up of lipids and proteins. Have you understood? Now students, what is the function? What is the main function of endoplasmic reticulum? Rough endoplasmic reticulum, ribosomes are attached, help in the synthesis of proteins. Smooth endoplasmic reticulum is also there. Okay, because it manufactures fat, lipids. Okay, so but in general, what is the function of endoplasmic reticulum? This endoplasmic reticulum is actually the membrane bound tubes. So this endoplasmic reticulum, it serves as a pathway to transport materials. Now if there is it is in the form of tubes. So what is happening? It is running in the cytoplasm. So what happens? It helps in the transport of material, or it serves as a pathway for the transport of material. Okay, if that is the endoplasmic reticulum. Now we come to another structure which is called as Golgi Apparatus. What it is called? It is called Golgi Apparatus. Children, this Golgi Apparatus was described by Camillo Golgi. Who described? Camillo Golgi. Now, what is Golgi apparatus? Golgi apparatus consists of tubes which are parallel to each other. It consists of tubes which are parallel to each other. In endoplasmic reticulum, what was there? There were tubes, and those tubes were not parallel to each other. Okay, but here the tubes are parallel to each other. You can see in the image 1, and you can see another detailed image also, which is shown image 4. So can you see, children? in image 1 also you can see the Golgi apparatus. What difference? Actually the endoplasmic reticulum and Golgi apparatus will look similar only. What is the main difference? That in Golgi apparatus the tubes lie parallel to each other, but it doesn't happen in the endoplasmic reticulum. So, and these are called as cisterns. What are cisterns? The tubes which lie parallel to each other, they are called as cisterns. You can see in the image also, the image 1 and also in image 4. Now children, what is the main function of Golgi apparatus? This Golgi apparatus that is connected to endoplasmic reticulum. But what is the main function of Golgi apparatus? Golgi apparatus, it is present in plant cells also, animal cells also. Now whatever is prepared by endoplasmic reticulum, that is transported outside the cell or within the cell by Golgi apparatus. Have you understood? Because in Golgi apparatus there are tubes which are parallel to each other. So whatever things manufactured by endoplasmic reticulum, because you know endoplasmic reticulum manufactures proteins also, fats also, lipids also. So whatever thing is manufactured by endoplasmic reticulum, how it will be transported outside the cell or to some other place in the cell, it is transported by Golgi apparatus. The things are packed and they are transported by Golgi apparatus. Okay. Now the function of Golgi apparatus is storage, modification, packaging, and also it helps in the formation of lysosomes. Now we come to another structure, that is lysosomes. Now children, these lysosomes are present in the cells only, and you refer to image 1. They are the organelles which help in the disposal of waste material. It disposes of or it throws out the waste material which is present in the cell, or which like food, some food, or some you can say the bacteria. So actually it cleans up the cell. How? It digests, by digesting it, because lysosome has got very powerful enzymes, and when these things, the foreign bodies, they enter in the cell, they come in the lysosomes, and then these powerful enzymes digest them. Digest them means break them, okay, and then they are thrown out of the cell. So actually it cleans the cell. Now children, sometimes what happens when cell gets damaged, suppose cell is damaged, so sometimes what happens, the lysosomes burst. Now when the lysosome will burst, what will happen? Digestive enzymes will come out, and those digestive enzymes, they digest the lysosomes only. Okay, it digests, you can say digest its own contents. So that is why lysosomes are also called as suicidal bags of the cells. Okay, why lysosomes are called as suicidal bags? Because sometimes the powerful enzymes, when the cell bursts, cell is damaged, the lysosome bursts and they digest its own contents. Now next structure or my organelle is mitochondria. Now children, you can see image 1, that is of animal cell. You will see mitochondria, and you can also see image 5. Now mitochondria is called as powerhouse of the cell. What is powerhouse actually? From where we get energy, that is powerhouse. So mitochondria is powerhouse of the cell, means it releases energy. Okay, it releases energy in the form of ATP. Whatever, see, cell organelles perform so many functions. Okay, so for that it needs energy, but from where that energy comes, it comes from the mitochondria. And in which form ATP, that is adenosine triphosphate. In detail, how it comes or how the energy is released by ATP, you will study in class 10. For the time being, just remember that energy is stored in the form of ATP, and it is released also in the form of, it is released by the ATP for various functions of the cell organelles. That is why it is also called as energy currency of cell. What is called energy currency of cell? ATP. ATP is called as energy currency of cell, and mitochondria is called as powerhouse of cell. Okay. Now students, this mitochondria structure in detail also it is given in the image. You can see detailed structure also. Now students, this mitochondria, I told you most of the structures of the cell are membrane bound. So mitochondria has two membranes. It is surrounded by two membranes, the outer and the inner. The outer membrane is porous. Porous means it allows the material to pass through it, but the inner membrane is highly folded. You can see. This mitochondria, it has its own DNA, which is the other structure which has its DNA, which contains DNA. Anyone can answer? You can use your microphone and answer. The structure which contains DNA. Okay. So another structure which contains DNA is mitochondria. It contains DNA also, ribosomes also. So if it contains ribosomes, it is able to make its own proteins. Okay. Now children, next structure which we will study is plastids. Plastids are found only in plant cell. Okay, you can see the picture, you can see the image of plant cells. Now there are two types of plastids. One is chromoplast, which is a colored plastids. Another is leucoplast, which is white plastids. Okay. Now this leucoplast, it is a white colorless plastid. It contains starch, oils, proteins. Okay. Now there is another plastids which contains chlorophyll, and those are called as chloroplasts. Have you understood? So the other portion we will study in the next class, but whatever we have studied today, kindly go revise it. Now I will ask few questions from you. Okay, you will use your microphone to answer the questions. Question number 1. Why mitochondria is called powerhouse of cell? Just write the answer in 1 minute, because these questions are very, very short. So don't take much time to write, and I can ask the question from anyone. Why mitochondria is called as the powerhouse of cell? Question number 2, which organelle is called suicidal bag of the cell? Or which organelles are called suicidal bag of cell? And why? You have to give reason for the answer. All the students will write the answers in their copy, and at any time from any child I can ask to send the photo. So the child from whom I will ask the question will answer the question using microphone, and other students will write the answers in their copies. Okay, so any time I can ask any child to send the photo of the answers through photo. So okay, all of you write the answers first, then I will ask the answer. Third is, in which organelle you find cisternae? In which organelle you find cisternae. Next question, what is plasmolysis? What is plasmolysis? Next question, what are eukaryotes? What are eukaryotes? Next, what are genes? Okay, so you can write the answers. All of you can write the answers. So if anyone is having any doubt, you can clarify now also. You can clarify in the evening also. Now question number 1. Why mitochondria is called powerhouse of cell? Nandini will answer. Or Nandini, you just write the answer or take the photo and send the photo. Nandini, you take the photograph and send it to me, WhatsApp it to me. Now question number 2. Which organelle is called suicidal bag? Konki, you please answer by using microphone. Question number 3. In which cell organelle you find cisternae? Priyanshu, please answer by using microphone. Other students, you write the answers. What is plasmolysis? Christina Mehdi, just send the photograph of that particular answer. What is plasmolysis? Okay, what are eukaryotes? What are eukaryotes? Rita Shri, tell me the answer using microphone. Next is, what are genes? Somiran, just tell me the answer using your microphone. By microphone, you can give me the answer of what are genes. Okay, now Pragyan, you take the photo of all the answers, okay, and send the photo to me through WhatsApp now. All the students will write the answers. Some of the students will answer using microphone, and from some of the students I can ask them to take photo and send the answer of 1 or 2 questions, or some will send the photograph of all the answers. Okay, so children, just please go read the chapter nicely, thoroughly. If you have any doubt, please clear it. You can clear during the class hours also, after class hours also. Thank you.